To better understand how DNA works as a language, I'm going to trace the process from reading a DNA segment to the production of a protein to be used in the cell. To simplify, I will leave out explanations of mechanical devices required to make this process happen, along with many other details. You are probably familiar with the double helix form of the DNA molecule. Let's start by looking at one small segment. Just as computers run on machine language, which is base 2, that is zeros and ones, basically on or off, cells run on DNA language, which is base 4, that is C, T, A, or G. It is important to know that though only one side of the DNA will be read, C is always coupled with G, and A is always coupled with T. To read the message, the DNA is split into separate strands. A copy of the removed side is replicated with parts that become a messenger RNA, and then the DNA is reassembled. Note that the T is replaced with a U molecule, making it distinct from DNA, but still representing the original message. A passcode is attached to the messenger RNA. This is a one-way ticket through the nucleus membrane into other parts of the cell. The entire messenger RNA is actually a much longer polymer than we have pictured so far, typically consisting of hundreds of letters, maybe over 1,000, all grouped into threes. Each group is to be read as one codon of information. This is comparable to a computer being raised from a machine language to a higher level language, such as Fortran. Each of the 3 nucleotides in a codon has 4 possible letters, which means there are 4 times 4 times 4 equals 64 different codon possibilities. If DNA was stuck with a binary code like our computers, it would take 2 times 2 times 2 times 2 times 2, almost twice as many iterations to achieve the same 64 possible combinations. These 64 codons correspond to 60 different transfer RNA molecules, each carrying a specified amino acid for the messenger RNA to assemble into a molecular chain. Plus, there is 1 start signal and 3 stop signals. Once the amino acid is connected, the transfer RNA is dismissed. Once the amino acid chain is complete, the messenger RNA is dismissed and the chain is folded into a specific protein component that the cell needs. In this case, a polypeptide chain of 594 amino acids are folded into the B'1 protein subunit, which is returned to the nucleus to be joined with the butyrate response factor 1 and TATA binding protein to form the transcription factor 3b assembly. That is then added to a molecular machine called RNA polymerase III. This assemblage is one of the devices that travels down the DNA and stops at a designated spot, separates the DNA into 2 strands, copies out the messenger RNA, and reassembles the DNA ready for future use. Proteins like this are required to unzip DNA and release its message. We know of no other way to unlock this information, and DNA is necessary to produce a messenger RNA. We know of no other way to produce RNA that contains a message, and the messenger RNA is necessary to produce the protein parts of the molecular machines that read and process DNA. We know of no other origin of such proteins. This is an interdependent, that is, circular system that has no possible explanation for coming into existence one step at a time. For example, why would a protein come into existence by some simpler way and then later require RNA for its production? All parts must be in place at the same time or nothing works. And if nothing works, all parts present are selected out generations before other needed parts could be expected to evolve. Just like Escher's famous hand drawing, the only way we know such a system could come together is all at once through an external master plan produced by an intelligent designer.